All set. Hello, hello. My name is Shane. So just to kind of reiterate here, we have been open since about 2014. We were the first company in the United States to offer genomic-based diagnostic testing. So since then, we've really been able to expand what we're able to offer to patients. So our genomic unity kind of consists of a genome backbone, both for the patient and for the parents. And we're able to carve out gene panels as well from genomic data. So we're able to provide as much data for our patients. Our ear site is we're expanding into reproductive genetics. So our talk today will really impact how we're able to use direct amniotic fluid for whole genome testing. And our onco is our cancer product line, and Genomic Inform is for healthy individuals. So as we're all aware, exome data is pretty common right now in industry. We are able to capture way more with genome, as everyone knows. And so our end-to-end solution is, we're able to take samples, numerous different types, blood, saliva, fibroblasts, muscle tissues, and a host of others. We perform whole genome sequencing. We have a proprietary system for bioinformatics, as well as a diagnostic console. And we have all of our interpretation in-house. So we have about 40 board certified genetic counselors and PhDs who do all of our interpretation. And then we're able to give a diagnostic report to a patient. So today I'm here to talk to you about tagmentation technology in whole genome sequencing and how we're able to compare that with larger DNA inputs that have previously been needed for diagnostic use. Just to kind of everyone that helped here as well, Adelise is our lab supervisor, wizard with a pipette. Near is our head bioinformatician. Raywin is our quality control wizard. And Ayla and Alex are our VPs, the first of lab operations, and the second is of our product development. So the real advantages of tagmentation are our low DNA input. So we're able to see that at varying inputs, we have high sensitivity, high specificity, and high positive predictive value. We sequenced about 50 samples consisting of 5 Coriels, NA12878, NA24385, NA24381, and 2 more. And so we're comparing them with reference VCFs from well-characterized for these well-characterized samples. And we see that at least 50 nanograms, we're getting consistent sensitivity, specificity, and positive predictive sensitivity and specificity. The positive predictive value is slightly lower. So we do prefer to go 50 nanograms and above. If there is a dire situation, we can go for 25. The second is it's a significantly faster workflow. So tagmentation itself is about 90 minutes library prep time compared to probably about 2 days as previously used. There's a reduction in sample recollection. So about 2% of our samples need to be recollected either due to low DNA quality or low quantity. So half of our recollections are due to low quantity. So here we'll able to proceed and get patients' reports quicker. This workflow is also adaptable to automation, which is huge in terms of scaling. Right now, we just opened up the lab about 4 or 5 months ago. So this is, we're working into automation. We see very high quality data coming out of this, which is comparable as well to the previous kits we were using. So right now, our process will analyze small sequence changes, structural variants, high complexity regions such as SMN1, SMN2 copy numbers, short tandem repeats. So we'll analyze about 26 short tandem repeats across CTG, CAG, GG, CCC. It's out of about 50 total neurological known pathogenic short tandem repeats. And we also are looking at mobile element insertions and uniparental disomy and mitochondrial variation. So just to kind of walk through here, we ran 145 samples spanning the different positive controls. So these were all from Coriell, and we were limited in some aspects, especially with mitochondrial deletions. They're not very easy to get a hold of. So small sequence changes, we see no issues here. We detected everything we were looking for, structural variants as well, short tandem repeats, mitochondrial variants, and the mobile element insertions. Again, mobile element insertions, we were only able to find 4. For SMN1, 2, we sent out our samples for MLPA testing to confirm the copy number, and we are developed a algorithm for looking at the copy number for the region. And so, okay. Also look at uniparental disomy. All right, so for analytical validation, we used the same reference VCFs. We see a 99.857 for single nucleotide variants. We start to see it going down only to about 96.0 for 16 to 50 base pairs, delins. We do obviously have the limitation in terms of smaller copy number variations under 300 bases. But as we get larger, we're actually we are detecting more than Illumina's out-of-the-box Dragon solution right now. So that's something and we also have a much higher positive predictive value. Short tandem repeats, we did not have positive analytical controls. Mitochondrial variants, we did a dilution of 15 different concentrations, and we see that we're able to detect heteroplasmies down to 2% reliably. And mobile element insertions, again, these span between 50 to 300, 300 to 1,000, and 1,000 to 10K, depending on the variant. And So we do see, again, this 83.5% sensitivity with a high positive predictive value. Just to get a look at our heteroplasmy, so we combined two Coriell samples at different concentrations, and we diffed the VCFs and looked for the reference and alt reads and made a percentage. And so we came up with, we have a 99.4% sensitivity. For SMN1 and 2 as well, we see good separation. We've previously seen some overlap with previous kits. We ran it for about 3,000 cases. So this is improvements. We're still going to be confirming anything that comes through, as we do with everything else. And lastly, the real impact this has for our patients is that we're able to do direct amniotic fluid. So that's something we have some preliminary data on now, where we don't see any dip in sensitivity and specificity. When we compare cultured, sequenced at 1 microgram, and direct amnio sequenced at 300 nanograms. We're also looking to expand into other neonatal and fetal testing, and buccal swabs is a big thing for a lot of patients. They do not, sometimes it's difficult to get blood, especially out of newborns or patients with, any sort of, who might have intellectual disabilities. So, any questions? Okay.
Okay, so you are actually responsible for the part that is related to rare disease diagnostic, right?
I work under Alex Kaplan.
Yeah.
So I was the laboratory operations manager, and I've been here for about 3 years with the company.
Yeah. No, the question was, so do you have any specific protocol for the diagnostic of rare disease?
Do we have specific protocols for?
Yeah.
So we do have specific protocols based on specific genes. The protocols can be 100 or 200 pages long for our variant scientists and genetic counselors and review board. So it's heavily dependent. So we confirm every short tandem repeat either through triplet repeat PCR, Southern blots. So we have a workflow which pops everything up that needs to be confirmed early in the pipeline, so that it can be sent out by the time it's being reviewed. So we're able to cut down turnaround time there. And I'm not familiar with the particulars of the interpretation very well, but I can point you to Alex, and we can follow up.
Okay, thank you.